just our sort of orientation material, and then we'll have this recording, which is going to introduce us to this wonderful world of mycology, which really we are in this course going to dissect a little subsection, a little sliver of this much broader world of mycology that for me personally tends to continuously spark my curiosity, my interest, and the relevance I see of how fungi are playing out in the world is really, I think, interesting in the context of the modern time. So before we get into how we're going to grow some specific mushrooms in some specific ways, what we want to do is really look at the big picture, what these organisms are and how they fit in to the greater world. So mushrooms are part of kingdom fungi. Kingdom fungi is an estimated 5 million species in the world. And mushrooms are just a small subset of that, where we look at fungi that produce these microfilaments we call mycelium, and that actually produce a fruiting body that we call a mushroom. But within this kingdom of fungi, we have yeasts, we have molds, and we have mushrooms, and we have lots of other non-fruiting fungi. The vast majority of mycorrhizal fungi that we'll talk about a little bit. So those are fungi that partner with trees and plants in the environment. These mushrooms don't actually produce a fruiting body. The vast majority of them, you'll never see above ground any evidence of them, but they are there in the soil. So keep that in mind, that when we talk about fruiting fungi, and when we talk about mushrooms, this is a small subset of a much larger kingdom. So if you're enjoying a fermented beverage at the current time, or plan to do so tonight or in the future, that's coming from a fungal organism that's doing that fermentation. We think about things like tempeh. Tempeh is a traditional Asian preparation of soybeans, where a fungi is actually inoculated into the soybeans, and it partially digests those soybeans, which then allows us to better digest the beans when we consume them as tempeh. We have, your common bread mold would be in this kingdom, and it certainly is not something we desire to cultivate in most cases on our edible foods, but is something that's part of this kingdom. So it's a really fascinating group of organisms that kind of spans the gamut from really wonderful to sort of some of our most problematic out in the environment. So somewhere in the range of 1.5 to 5 million estimated species of fungi are out there in the world, in the living environment. We've only named about 5% of them. So we have a long way to go. And certainly when we compare it to plants, to animals, to other forms of science and botany and things like that, mushrooms are just far, far behind in terms of what we know, who's studying them, what we're learning about them. There's new species being discovered and named every single year, and just continues to grow. If you were to walk around the woods in most parts of temperate North America, you might come across one of 10,000 species of fungi that specifically form a fruiting body. So when we often call those mushrooms, those fungi that form that kind of distinctive fruiting body. It can be soft, it can be hard, it can have gills, it can have teeth, it can have all sorts of different shapes and forms, but generally speaking, when we see something that is that reproductive part, we call that a mushroom. These are rough numbers. Out of those 10,000 species, maybe half of them, or probably half of them, are just kind of inedible woody conchs and shelf mushrooms that we see out there in the woods. Generally, like 25% or so are edible, but they don't particularly taste amazing, but we might eat them in a pinch or really add a lot of butter to get them to taste good. About 20% make you sick. About 4 or 5% are actually incredible, delicate, sort of gourmet, really high-end mushrooms, and maybe 1, maybe 2% can actually kill you. So what's important to notice here is the vast majority of mushrooms are sort of neither incredible nor are they poisonous, but in most cases they are inedible. Now, it doesn't mean we don't need to do good ID, and we'll talk about that a bit tonight and in future classes, but know that the mushrooms that are out there, there's a very small amount of them that actually will kill you or make you even sick. And when we intentionally cultivate mushrooms, what we're doing is we're kind of taking that variability, that unknown that we have of looking at mushrooms in the woods, we can actually bring them into intentionally cultivated systems and pretty much eliminate any possibility of contamination or some other issue fungi, which we'll talk about as we go here. Kingdom fungi is on the tree of life is closer related to the animal kingdom than it is to the plant kingdom. So we are actually more closely related to fungi than we are to plants. And what's important to make note of is that both us and fungi are heterotrophic, so we digest food. We're not creating our own food from photosynthesis, right? That's the main difference when we look at fungi and animals versus plants. The biggest difference between us and fungi is that we digest the food in our stomachs internally with enzymes, and fungi do it sort of outside. They excrete enzymes from their cell walls out into the environment, and they digest foods, and then they bring those foods in once they're digested into their cellular structures. And so it's a really interesting thing and it's relatively recent that scientists really understood this, because for a long time it was understood that fungi were just some kind of weird mutation of plants because they didn't move, they didn't have any locomotion, and so they got kind of lumped along with plants. But as scientists looked more in detail, they realized that fungi didn't need oxygen in order to survive. Or excuse me, they didn't need carbon dioxide in the same way that I'm sorry. They breathe oxygen and respire carbon dioxide, right? They're like us in that way. So we all need those things, but fungi did not need carbon dioxide in the same way that plants needed, in the same concentration. They also didn't need sunlight in order to grow and to reproduce. And so that was the first kind of spark to realize they weren't plants. And as time has gone on, research has gone on, they've realized they're very closely related to us in the animal kingdom. They spread through their material. They actually grow through their food, which is a big distinction from animals to fungi. Initially, we call this growth pattern sort of hyphae. As these hyphae merge and expand and grow further, we might call those mycelium or mycelial masses. And this is actually the fungal organism itself. If that body produces a mushroom, then that becomes the sort of fruit of that organism. And the reproduction cycle continues with that mushroom producing spores, which is really the sort of genetic information that's being put out into the world for the purposes of reproduction. And so, like I said, that they digest food in their environment with enzymes, and that's really how they go. So another thing is that unlike plants, what's interesting is that hyphae and mycelium grow multi-directional. They grow they don't just grow a root down and a shoot up, they can grow in every direction through their material. And they can grow incredibly fast. So oyster mycelium in particular is very fast in terms of how fast it can grow through its substrate and actually fruit. So you can have oyster mushrooms fruiting in as little as 3 weeks from that initial mycelium growing to the actual fruiting mushroom itself. Other mushrooms can take years, and some mushrooms are not predictable, nor can they sort of be replicated as a cultivated mushroom. And we'll get into some of the nuances as to why. Unlike plants, unlike animals, mushrooms don't need a generation of reproduction in order to express sort of new traits. Mushrooms have a really unique morphology. And one analogy to think of with fungi is that as they encounter a new or unknown source of food, they often can excrete and create new chemical enzymes in order to break that food down. And they can do this without reproducing, so they don't need to have offspring, adapt to those different changing conditions. They can actually do it as they grow. This is sort of a remarkable characteristic of fungi that give them a really unique role in the world. This is why fungi are the original decomposers of rock material. They weather rock and they turn it into material. They're the original decomposers of wood and lignin. They can break down these really complicated materials. Researchers have found that mushrooms can break down plastic, all sorts of different things in their environment, and it's about learning and adapting, and they can do this incredibly well, incredibly effectively, and incredibly fast. So morphology is a really fascinating part of fungi. So of course, with our fungal friends and our mushrooms, we have lots of good, wonderful companions. We have this fella here who's kind of overwhelmed with his haul. He's got a maitake, or a hen of the woods, there in that picture. This is also known, taki means mushroom and mai means dancing, and so the maitake is the dancing mushroom. And because usually you find these in large quantities and you find them in the woods and you cannot cultivate them too easily. You can do it indoors on sawdust, but you can't really reliably cultivate them outdoors. But maitake is something you find in the woods in the fall, and you do a little dance because it's such a fun mushroom and a delicious and highly medicinal mushroom to find out there in the forest. We have the lion's mane here on the top right. Really wonderful mushroom, both edibly and sort of medicinally. We'll talk about how to grow that in the woods outdoors this class. It's known as the memory mushroom, and so it's got a ton of medicinal properties when it comes to stimulating brain function, cognitive function, restoring nerve growth, endings, things like that. And then, of course, we have blue cheese, which is a fungus. And lots of those high-end cheeses are actually a fungal mycelium that's colonizing the cheese and actually creating that sort of interesting flavor, right? So we have the good, we have the bad in our fungal kingdom. Some pictures here of course, foot fungus, athlete's foot, nothing pleasurable about that. Jack-o'-lantern mushrooms. These mushrooms are one of the more common ones that people try to eat and get sick from. They're not chanterelles, and chanterelles look very different than these, but if you saw an orange mushroom in the woods, you might get excited and think it's a chanterelle and join folks who have made that cause severe vomiting for. And then pathogenic fungi, of course, as well, like fire blight down here on the left corner, or the beech bark disease here in the right corner. So lots of fungus that, we could say do bad things to trees or create challenges for crops and for our forestry. But as we look at the big picture, a lot of these pathogens really also help with the natural selection process, with improving the overall health of the ecosystem. So I think science is starting to really appreciate the role of it. Ugly, we have the really intense fungi, the ones that get the all the stories, and so I think often cast a dark shadow on some of the other we have millions and millions of different species, but sometimes those few that get the, oh my gosh, can you imagine this mushroom that's called the death angel, right? This is going to kill us, and so all mushrooms must be bad. Well, there's no mushrooms that show up in solitary growth patterns, or in this case in pairs in the woods, that are pure white, that have that chalky white stalk there you can see in the photo, that have that really drapey cape-like ring that's left when the mushroom opens and separates from the stem, that is literally white on the base, on the stem, on the cap, that has a vulva at the bottom. You can see the little lump or the Easter egg that these mushrooms actually popped out of. We call that a vulva. I mention all these characteristics not so that you know them, but so you understand that if I strung all those things together, there'd be no choice edible mushroom that we'd ever mistake for this Amanita, which is the death angel, right? One of the ones that we hear a lot of people being very fearful of, for good reason, because this one would kill you. Likewise, the chestnut blight being one of the worst fungal diseases that knocked out the American chestnut by and large in the eastern U.S. Certainly one of the more devastating and rapidly forming blights and rapidly spreading, but not what we tend to see as the way pathogenic fungi sort of work their way through the world. So, fungi are also amazing in that they create these symbiotic partnerships. They're always out there working with other good fungi. So if you've ever had kombucha, this has really become a craze in the last few years. This is a symbiotic yeast or fungi and a bacteria that are actually creating that SCOBY, that mother that does the fermentation. A lichen is a fungi who's partnered with an algae. So the algae is able to photosynthesize and actually pull that energy in from the sunlight, and then the fungi itself is able to excrete enzymes and actually decompose the rock and harvest minerals. Essentially, it's a mining agent. And so the two of those together can create these lichens, which are some of the most diverse and resilient species that we know of on the planet. Far before humans ever cultivated fungi, we have lots of different insects and critters actually actively cultivating fungi in their homes, both for food purposes, for protein sources, and also as a medicinal compound. And so fungi have this really important role to play, and have certainly partnered with many different organisms in order to succeed, in order to spread. And colonies have been, bees, I should say, in Africa that have been cultivating mushrooms for several thousand years. So we might have something to learn from these critters. Bees also harvest, cultivate, and work with fungi out in the world. We're going to break down some of the different types of fungi that are the main ones we think about, and why some fungi might be easier to cultivate than others. But it's important to remember from the outset that all fungi are essentially soil builders. Their role in the environment is to break things down, capture and cycle nutrients and water, weather rock in ecosystems, break that down and harvest the nutrients and the minerals from that material. And so all fungi really perform that role and are critical. And so if we're interested in growing mushrooms, great, but we also need to think of, if we're interested in cultivating plants or in supporting healthy pasture or healthy livestock, fungi actually show up in those systems too. They're ultimately tied to almost everything. And this is really getting at, for me, what has started off with just wanting to grow some delicious mushrooms and really has blossomed into a much larger understanding of what's out there. So within the decomposing mushrooms, we have mushrooms that sort of prefer and desire different types of food. And so some fungi are what are known as white rot fungi, and these are the fungi that love fresh wood, that love cellulose. We sometimes call those the primary decomposers. Excuse me, not cellulose. They love the lignin in the wood, so they're actually the first types of decomposers on the scene. They break down the lignin in the wood and they use that as their food source. And so you can grow these mushrooms on this wood for a long time, and then they're spent, but there's still a lot of cellulose left. And then we have a whole nother suite of organisms known as brown rot fungi, such as the chicken of the woods here, that feed off the cellulose. And so one log different food sources, several fungi. And this is the way that nature figured out to deal with its waste problem right out there in the environment. And so, you know, what we know from this and how this applies is that a lot of the easy mushrooms to cultivate, oysters, lion's mane, shiitake, are white rot fungi. They decompose wood, and we can set that arrangement up for them. Chicken of the woods, a brown rot fungi, is much harder to cultivate. And partially it's because it's much later in the decomposition cycle. It's a secondary decomposer, and there's other factors, there's other things going on in that wood by the time it's reached that stage. So it's a little bit harder for us as growers to set up those conditions and make sure that they work well. So mushrooms break down wood, they create soil in that way. But another set of mushrooms that aren't decomposers per se, but do a lot of the important work, are mycorrhizal that have done and have shown to do a lot to transfer a lot of water, nutrients, minerals, and even information from tree to tree. If you haven't checked out the work of Susan Simard, we have some videos in the links this week. She's done a lot of work with mycorrhizal fungi in British Columbia and the Pacific Northwest, and she's found that the roots of trees are connected to each other underground in these old forests through networks of mycorrhizal fungi, many of which will never produce a fruiting mushroom, but nonetheless have worked out these relationships where they trade essentially nutrients and water for the photosynthetic carbohydrates that are coming off of the trees. And these relationships are millennia old. Some trees have relationships with only one or two species of fungi, and others have relationships with hundreds and hundreds of different species of fungi. It's said that it's kind of a loose number, but somewhere between 75 and 90% of all plants have a mycorrhizal relationship. And so all sorts of amazing things happen. So mycorrhizal networks can store carbon and cycle it for long periods of time. It's actually the mycorrhizal fungi networks that do a lot of the carbon sequestering that we hear about in soils. Mycorrhizal networks can help move nutrients from a tree that might be abundant in them to one that's more deficient in them. Susan Simard's work has shown that there are mother trees, such as in this photo, that might actually share nutrients and minerals to younger trees, to its offspring, and they actually will preferentially do that in a forest. They'll find those offspring and help them along a bit more than the others. And there's just the stories go on and on about these kind of amazing relationships that are happening. Finally, we have mushrooms often thought of as vegetarians, as eating just wood and waste and things out in the soil, but we also find this interesting property where some fungi will actually prey on living things. And so here's a picture, in this case, of wine cap stropharia, which we'll talk about growing in the course, and it is lassoing and consuming this nematode in the soil. And so mushrooms are not necessarily vegetarian. They may be omnivorous at the least, and can have roles to play in soil health. If we imagine the potential challenges that nematodes can play, well, fungi can help offer a competitive force into these kind of pictures of things. So lots of different ways that mushrooms play out in here. I'm gonna keep rolling here with things. I see some folks are mentioning some sound issues, which I apologize for that. Hopefully it's intermittent and it's not too bad. If you again try the phone if you haven't tried the phone, if it's not working. Also, you may have to review the recording if it gets really bad, but there's not a lot I can do on my end at this point other than wish you well with those modes of things. So I'll leave that for that. I do see there's a couple questions, which I'm also going to leave towards the end of this section, if you don't mind, so we can keep rolling here. So we're going to talk a little bit about the mushroom life cycle and get a little bit more specific about the types of mushrooms that we might want to cultivate and grow and how we could interact with them tonight. Again, we're kind of keeping things big picture. Starting next week, we'll just keep drilling further and further in. We have to talk about fungi as a whole, then we have to talk about the forest, then we have to keep talking about specific ones we can cultivate. So if you're ready to cultivate tonight, you have to wait a few weeks because we're going to keep working on some of this background information that's actually really important to understand if we want to grow mushrooms successfully. So what's important with the mushroom life cycle and mushroom biology to understand at this point is that different animals, and it's important to understand where we kind of fit in and how we work with fungi given this life cycle. So we've talked about the mushrooms being the fruiting body of some types of fungi that create mushrooms. In this case, we have an oyster mushroom growing through the life cycle. So a mushroom's purpose, whether it has gills or teeth or something else, is to produce spores. Spores are essentially like microscopic little sticky packets of DNA, just information. They don't have a protective coating like seed. There's no way I could pick up a spore out in the environment and stick it in my pocket and travel halfway around the world and then replant that seed. That just doesn't happen. I could do that with a plant seed, but I can't do that with a spore. So a spore is just a kind of sticky, and actually a single spore you cannot see with your naked eye. And so the reproductive strategy of mushrooms has been to produce millions and millions of these spores, kind of shoot them out into the world and hope that they land on a suitable food source. So the vast majority of spores don't make it, essentially, is what it comes down to. So you can see the spores, the zoom in here is just showing what an interior of the gill walls might look like with all these cellular structures and the spores actually launching from each of these individual veins in the gills. The spores will hopefully land on a food source, and if they find something suitable, they can actually germinate, just like a seed might. And germination really just means they start to kind of hatch out and grow a little filament of hyphae. In mushroom reproduction, you need at least 2 compatible mates in order to have those initial strands of hyphae actually merge and grow and continue to turn into a specific mushroom. So they have to find that, and it's not as simple as like male and female in that sense of reproduction. Some mushrooms have thousands and thousands of different combinations of sexes in order to have that happen. So that spore phase is actually quite complicated and is not something that we as cultivators, generally speaking, engage with. What we generally do is get to the later stage of growth where those hyphae have grown out in sufficient mass and become mycelium. Mycelium is generally introduced in cultivation to a substrate, which would be a food source that we've crafted intentionally so that mushrooms can grow on it. So that's substrate. So we take the mycelium, we introduce it to the substrate, and then we nurture that substrate through the growth phase, where the mycelium grows entirely through that substrate and then begins to fruit. So in the case of logs, that might take 6 to 12 months before that mushroom's ready to fruit. And we need to make sure those logs are kept in the shade and well moist. And there's lots of things we can grow mushrooms on. In this course, we're going to focus mostly on the things that we find out in the woods and outdoors. After that substrate has fully grown with the mycelium, if the fruiting conditions are right, then we can have pinning or primordia, which is when those first mushrooms start to form and show up on the log. And then we, of course, have our mushroom, which is our final fruiting body. So spores and types of spore delivery really, morphology-wise, just show up as different ways that mushrooms behave and sort of demonstrate what they look like. And so the vast majority of edible mushrooms are gilled. The vast majority of fruity mushrooms are gilled and often are the hardest to identify just because there's so many differences in them. So I get hundreds and hundreds of pictures a year with gilled mushrooms that people want to identify, and in many cases I can't because they don't take a great picture and they don't show the top and the bottom and the side. They don't take a spore print. They don't do all these different things that are necessary for ID. But there's lots of nuances in the gills that can really help you understand how to identify them. So in this class, we'll talk about oyster, shiitake, stropharia. We'll talk about almond agaricus and namiko, and those are all gilled mushrooms that we can grow outdoors. Toothed mushrooms are less common, but there's a few species, and one of the ones that will grow in the course, we'll talk about that, is lion's mane, and it's a wonderful mushroom and very easy to grow, very easy to identify. Toothed mushrooms are some of the easiest to identify because there's just not that many of them. And then we have the polypores, which would be, instead of gills or teeth, we just have these kind of holes showing through the mushroom, and that's where the spores are coming out of. So some of our favorite edibles that we find in the woods, chicken of the woods, maitake, turkey tail, these would all be, polypore type mushrooms. Here we have some mycelium of red wine cap, right? This is the fungus that will produce mushrooms. And this is, as cultivators, what we're trying to start with, inoculate into new food sources, and then encourage them to grow and to thrive as they work on that journey to producing fruit. So I like the wine cap mycelium. It's very coarse in terms of its threadiness. You can see it's growing on and in all this kind of wood chip material that we have it cultivated on. Here's primordia. We could just simply call these little baby mushrooms. Primordia is a really important part of the growth cycle because it's triggered by the right environmental conditions. In the case of shiitake, which is what this is, we actually soak the logs in order to force them to fruit. The primordia pops up, and then we want to make sure that primordia doesn't dry out before it actually produces a mature mushroom, such as here, which could be, in the case of shiitake, anywhere from 5 to 7, sometimes as long as 10 days from when those primordia pop out to this nicely formed mushroom where the cap is actually separating from the stem and opening up, and we want to capture it and harvest it before it becomes a flat pancake. We want to keep those edges nice and curled, kind of like the lobe on your ear, your upper ear. If we have that kind of edge on our mushroom, then we're good to go, and that's the peak sort of harvest time. Of course, if we wait too much longer, the mushroom will go flat, and then it'll invert like an upside down umbrella, and then it'll rot off and be something altogether different and not desirable. So, one of the important things about spores is we're not actually going to do a lot of cultivation from them, and in most cases, folks, especially in the class here that are getting started with mushrooms, you're not even going to cultivate the mycelium. You're probably going to buy mycelium from a supplier and then inoculate materials. That's essentially what you're doing when you buy your mushroom kit, is you're paying for the folks at Field and Forest to do the initial cultivation and stewarding of this fungus. And by the time you get it, it's really in a usable form. It's ready to go. There's not a lot of things you have to worry about, the right food and supporting its initial growth. Spores still remain an important feature in a cultivator's toolkit when we talk about proper mushroom identification. So I want to just talk about a spore print. There is a video this week about making spore prints that I really recommend people review. If you're going to cultivate mushrooms, and if you're going to sell them in particular, you really need to demonstrate you have proficiency in identification, because you will be asked about it from your customers, from regulatory agencies. And a lot of people have phobias and fears about mushrooms, and so you need to reassure them that you know what you're doing. So a spore print is simply when you take a mushroom, you take a piece of aluminum foil, or you could take a black piece of paper and a white piece of paper, you put the fresh mushroom gills down on the paper, and you capture this beautiful imprint of the spores that drop over generally from anywhere from like 2 to 24 hours, depending on the mushroom. So I generally just put them down, leave them overnight, and then check it the next day. And that's really all you're doing. And that spore print actually can turn into quite a beautiful piece of art. I know some folks that are full-time spore mushroom print artists, but for identification, the color and the shape of that print can really be beneficial as we look at identification. And each mushroom has a unique spore print, and it's sort of like a fingerprint, or it's a unique signature of that species. And it really becomes critical when we talk about identifying the difference between certain mushrooms. It is not the only thing we use, but it's one of the most important. So for instance, log-grown shiitake, we're going to focus a lot of that on the course. It has a white spore print. A mushroom I would be concerned of misidentifying shiitake for would be Galerina, which will kill you. Galerina has a brown spore print. So just on that information alone, if I wasn't sure, I could take a cap of fresh mushroom, I could do a spore print. If it's white, I'm in good shape. If it's brown, I'm not. But of course, like with any identification, we don't want to just use one characteristic. So it's not just the spore print. What's the shape of the cap? What's the color of the cap? Is there a ring around the stem? What is the shape and differentiation, or the way the gills are spaced? What's the difference in the cap versus the gill color? You know, these all things are important in identification. So as with any good idea, we have 3 to 4 to 5 different pieces of information that we collect in order to make sure and differentiate one species from another. I will tell you that shiitake and Galerina, one of the most important features is that shiitake is a white rot fungi that grows early on in a log's decomposition life. Galerina only shows up late in a log's decomposition. So it is a brown rot fungi. And that white rot fungi means that if you inoculate a log with shiitake, I can almost guarantee it's going to come out with shiitake mushrooms. And if any other fungus fruits out of it, it's not going to look like a brown cap and stem mushroom with a light sort of off-white stem as we have with shiitake and Galerina. The biggest way you could misidentify a shiitake for a Galerina is if you grow shiitake on your log for 2 or 3 seasons, you toss that log in the woods, and you toss that log in the woods at the end of its productive life, right? So 3 seasons after you've produced mushrooms, you say, all right, this log's done, I'm going to toss it off into the woods. And then you happen to walk through one day and you see some kind of brownish looking mushroom that kind of looks like shiitake, and you don't think about it because, oh, this is a log you cultivated, and you pick those mushrooms, you put them in your basket, and boom, you eat them later and you're in trouble. So, you know, all this to say is that context matters in addition to things like spore print and key ID principles. And we'll dig a little bit deeper into ID in future classes, but, you know, just keep in mind spore prints. It's something you should start doing with fresh mushrooms just to get a practice on it. And before you start selling mushrooms, before you start consuming, you know, verify with a live human that you are eating the thing you think you're eating. So when we talk about domesticating mushrooms, when we talk about actually growing them, we're sort of skipping the process of sexual reproduction. We're not often cultivating mushrooms from spores, although it does happen occasionally. Mostly what growers are doing, or spawn suppliers, mushroom suppliers, are doing is taking a wild specimen and basically taking a cutting of that, taking a piece of it and growing more mycelium from it. So mushrooms have this amazing ability. You can take a little piece of that lion's mane there that's labeled wild, and you can take a little cutting of that, put it on a petri dish, as you see in the second photo there. That'll grow out onto the petri dish material, the agar or starch that you put in there, essentially a little bit of sugar to get it started. From there, you can take little pieces of that and you can inoculate sterilized grain, which is what this is shown. From there, you can inoculate salt. you can expand it even further into these sawdust bags, which are actually substrate that the company is designing to sell to you, so you can inoculate logs and create and grow more lion's mane. So the reason that most of us don't do this is because other than the first picture in this and the last picture, all of this is done in a very clean, sort of sterile environment, kind of like a lab. So if you happen to have a lab in your basement or something accessible, that's great, and you could do this. But for most of us, it's cost effective to outsource this, just like it's often cost effective to buy vegetable seeds from a good company. So spawn suppliers will provide you with this material so you can do your inoculation. And at least when you're getting started, I'd recommend you start there. And maybe someday you'll do some of your own, but for most of us, we're talking about 50 cents, maybe $1 per log to inoculate. So it's very inexpensive to buy what we call spawn. Right. So revisiting our mushrooms, why grow certain species and not others? What are some of the reasons? And I've highlighted a few, but I want to just revisit this because we get these questions again and again and again through our website and through our extension program. So mycorrhizal mushrooms, let's revisit those for a second. There's a lot of mushrooms that people want to grow that are mycorrhizal. So they form this beneficial relationship with the tree or the plant. In this case, it's a white pine seedling. You can see there with the brown roots and then the mycelium that's colonizing around it, and is growing into the soil and actually expanding the tree's root system 60 to 75%. It's pretty remarkable. It's a great symbiotic relationship. But for us as cultivators, that's a relationship that's really hard, if not impossible, to set up. All the conditions, research has shown that there's bacteria involved in this relationship. There's specific soil conditions needed. And so to think that I can set up those relationships as a grower, I think I'm a little more confident in myself than I should be. That's a really hard scenario to set up. So if you came to this class hoping to learn how to grow morels or zucchini or boletes or chanterelles or truffles, these are mushrooms that are near impossible, if not incredibly difficult to cultivate because they're mycorrhizal, because they have a whole forest ecology behind their successful cultivation. There's a bunch of parasitic mushrooms out there, cordyceps being one that people are really getting excited about, mostly for its medicinal value. And there's a video in the lessons this week that talks about this sort of killer mushroom that essentially takes over the nervous and brain system of insects and then fruits out of the mummified bodies of this insect. And it does this for an ecological reason, but it's a pretty far out sort of sci-fi way to see fungi operating in the world. But parasitic mushrooms like this is again not a relationship that we can easily set up where we can have a host and a species. And so cordyceps are actually mostly grown indoors on sterilized media, and there's been adaptations through breeding that have allowed this to happen without that intermediate host, i.e. the insect. So parasitic mushrooms, again, uber complex, hard to cultivate. Things like chaga, often we get asked parasitic mushroom. It only grows on birch trees. It generally inoculates the tree as it's living and lives in the tissue of the tree for much of the tree's life. And then as the birch tree is in decline or dying is when the chaga starts to form its fruiting body here. And so, again, a very hard sort of relationship to cultivate, just to inoculate, bring two things together and have success. This is something that's formed over decades, if not longer, in the forest. Okay, so what we grow when we talk about cultivating mushrooms, we grow saprophytes. We grow the decomposers, whether they're brown rot or white rot fungi. We grow those mushrooms that basically use dead organic material as their food source. So not mycorrhizal, they're not parasitic, they're saprophytes. And as I mentioned before, the white rot mushrooms are generally the ones we have the most ease in cultivation. So I mentioned that we have, one of 10, maybe 10,000 species of mushrooms we could come across. What are we cultivating on our farms and in our gardens? We're cultivating maybe 5 or 6 species at most that we can reliably cultivate and have success with. And all of them are decomposing mushrooms. Not all of these are the same. Here's chicken in the woods again, just to show another one. This is one of the easiest mushrooms for wild foragers to start identifying. It's very distinctive. It's a polypore, and there's actually no polypores in North America that have poisonous characteristics. So polypores are almost all inedible. They're just kind of woody tasting, but things like chicken in the woods are this delicious if you get it fresh. That is easy to identify, but really hard to cultivate. So again, it's a brown rot fungi. It's a secondary decomposer. You can see the log here is pretty broken down. It's probably been sitting here for 8 or 10 years versus the shiitake log, which has been sitting there for just a few weeks before, or I should say a few years before it was at this stage of growth, right? So very difficult sort of transaction for us as cultivators to set up. So we mostly focus on decomposers, and we mostly focus on the white rot fungi. At the top of this presentation, I talked about our specialty mushrooms versus our kind of conventional mushrooms. So because the button mushroom industry is so large in the US, we have actually named a different group, everything but the button mushroom, as a specialty mushroom. So just so you know, button mushrooms, cremini, and portobello are actually all one species of mushroom. They're all the same. They are Agaricus bisporus. It's just different parts or phases of the growth. So you look at these button mushrooms on the slide, they're essentially closed mushrooms that have a closed cap. They haven't separated from the stem at all, and they've been starved of light, so they haven't developed any brown pigment. If you let those mushrooms mature a little bit more, start to separate up, and you'll see a nice brown gill set underneath, brown-black. And if you give it a little bit of light, that cap will turn brown, and then you have your cremini. And if you let that mushroom fully mature and give it a lot of light, you'll get your portobello. So this is all one species, and this is over 90% of what's being consumed in North America these days, so we have a long way to go. And we are not focused on these mushrooms. These are saprophytes. They are decomposers. They mostly grow in compost, but we're not focused on them in this course because of the sort of scale and hold that the button mushroom industry has on this one species. So at best, you might get $2, $3, maybe $4 a pound, versus the specialty mushrooms where we might be getting 10, 12, 16, or even $20 a pound. And so for a small scale grower, it's not really worth getting into the button mushroom industry. It's worth focusing on some of the other species that are out there. So next week, we're going to talk a lot about the forest and forest ecology and how that relates to mushroom growth, specifically the saprophytes that we want to grow in our mushroom cultivation systems. But the reason we like the woodland mushrooms, as maybe compared to how most mushrooms are grown, which are indoor production systems, is this is a relatively accessible way for more people to grow mushrooms. It doesn't require any fancy skills or training or facility in order to make it happen. It will, as we talk about next week, support management for forest health. So if you have access to a woodlot, this can be a really good way to tie in objectives you have for that woodlot to also the production of mushrooms. It's certainly a niche crop, and this is something that growers have been able to really fetch a premium for log-grown shiitake, for woodland-grown mushrooms. It's something that we see the market being interested in. And woodland mushrooms in particular have a very low investment in terms of material and labor for a pretty high return, whether that's your own sort of consumption of the mushrooms or actually selling them themselves. We focus on this because a lot of our work is supportive and encouraging of mushroom growers all over. But specifically, we started doing grant research back in 2010, and we were looking at mushrooms because we were looking at the reality that a lot of farmland in New York State and in the Northeast has woodlots, and these woodlots need incentives for them to be managed and stewarded into the long term. So we looked at other crops besides timber, besides wood products, and things that could bring an income for the landowner. So in New York, 21% of existing farmland is woodland, and it's often seen as low value and mostly used for some timber and some firewood, and there's no problem with that. But we wanted to increase and improve the meaningful products that one could grow off of their forest. And so this is where the mushroom project originally started. We worked with landowners, and a lot of them had this kind of scenario. And what we realized is that in this case, we have a maple woods here. This is at a stage in its life development that could use some thinning. We could actually remove some of the slower growing trees, and we could encourage the ones that are doing the best to really fill in the canopy and be the long-term overstory. And that the material that we're actually pulling out from that thinning event is perfect material for mushrooms. If we left these trees alone, eventually they'd shade each other out, and those smaller trees would die, fall to the ground, and they would be consumed by some kind of fungi. And chances are that fungi wouldn't actually taste very good. It would just be some kind of woody mushroom. So all we're doing in the process of growing mushrooms is we're inserting ourselves into this process, and we're thinning the woods to support its long-term health. And then we're producing this material, which is really high value in terms of its ability to produce mushrooms. But we try to enter the woods, and this will be the kind of underscore next week. You know, we harvest mushroom logs as a surplus of doing good forest management. We don't walk into a woods and just pick what any old logs are. You know, specifically just thinking about, well, I just want to maximize most number of logs to that overall forest health. So when I started mushrooms, it was all about thinning our woods for maple syrup production. We had a lot of this small diameter woody material, as you can see in the photo here, and for many years we were just using that as firewood. But when we started learning we can grow mushrooms on them, it became an additional revenue source for the operation. All right, and this is actually getting into next week. I'm just gonna show a couple more slides here, and then we'll wrap up for tonight. Yeah, let me go here. So we talked about our agaricus and our specialty mushrooms. Profitability-wise, for outdoor mushrooms, it's important to kind of say this from the outset. Log-grown shiitake are going to be our #1 sort of bread-winning species if we're interested in commercial production. We'll talk about another 5 species that you can grow. Those mushrooms can certainly be sold at markets and fetch a fair price. But the real reason that the shiitake is the breadwinner is because you can get a reliable harvest of these mushrooms, generally like from June through October in central New York. And so if you're further south than us, you're going to have an extended season. If you're further north, it might be a shorter season. But we can actually get a weekly harvest of shiitake all throughout the growing season, which is really what lends them to being commercially viable. The next step from that, often what we're doing a lot of work with now in our research program is doing oyster mushrooms. And we will talk about a way to cultivate oysters on wood. We do get good production from those logs, but we don't get weekly kind of stable production. And so often the next thing we look at is growing them on something like straw or sawdust. And that is something that can be done outdoors. We'll talk a bit about that later on in the course. But what I want to keep in mind at this point is that we have our mushroom species that we can grow, we have profit potential. And through our research and our work, these are the ones that really continue to rise to the top if that's your main goal for production. We've seen specialty mushroom production just rapidly increase, and demand is continuing to increase. So this is a very good crop, and the forecast for the future looks very good, especially as people are looking for alternatives to consuming meat, looking for different sources of protein, and just generally seeing mushrooms as a really healthy and medicinal type of food product. So it all comes back to the wood's health. Some of the mushrooms we're going to focus on this course, I mentioned log-grown shiitake will be the main one because we can develop a very robust and consistent production system. We're going to talk about lion's mane, the 2 different species that you can grow, and the methods that have, and the wood species that have tended to work best for those. We're going to talk about winecap stropharia, or winecap mushroom, which can be grown really successfully in especially areas on the farm or the homestead where you're already mulching something with wood chips. These winecap can be integrated right into them, and they actually like to grow also around plants, and so in other plantings and other production systems they can do quite well. We'll talk about growing oyster mushrooms, and if you decide to follow along with the kit, we'll even grow some, and they should fruit before the end of this course. We'll grow them on toilet paper, just as an example of what can be done. Here's oysters on treated media. In this case, this is on a straw log, a synthetic log. We'll talk about namiko, which is a really wonderful mushroom that can be grown on black cherry and sugar maple. And it's a slightly different method. You can see here the logs are inoculated and then they're actually buried in the forest in order to produce these gorgeous mushrooms. So we'll go through these with the different species and the different steps in the process, but it's all about, for our work as cultivator, which we'll start next week. We're taking that fresh log, we're inoculating it with the mycelium. We're having some stage of colonization, which is really where the mycelium starts from those points of inoculation and spreads throughout the material. And then we have a period of time where we're doing fruiting and harvesting. And so these 4 steps are going to be something that no matter what you cultivate, you're going to be working through. But the types of substrates or the foods, the procedures for inoculation, and then the steps for managing them through sort of 3 and 4 there are all going to be different depending on the species that we walk through. Okay, I think that's going to leave us for tonight. We talked about the website. That this slide here. I'll mention that really great starting book for mushroom farming that I recommend everyone check out if you haven't already is Organic Mushroom Farming and Microremediation. This is a book by Trad Cotter, which really I think gives a nice overview, expands upon some of the concepts we've talked about tonight overall with fungi, and then looks at a lot of different ways that fungi can be integrated into productive systems. I wrote a book with Ken Mudge, who I originally learned with and worked for at Cornell, doing some of the research that we'll talk about when we look at log-grown shiitake in particular. And when he retired in 2014, we wrote a book together about farming the woods in general. So not just mushrooms, but other nuts and fruits and crops that can be grown or cultivated in the forest. But there's a whole section there on mushrooms that mirrors a lot of the stuff we talk about in the course, just in really a different format. I'm running it up against time. I apologize. I went a little over tonight, but that often happens on the first night when we have all that logistical fun stuff to to review with you all. So I'm going to stop the recording, and I'm going to hang out. And if you have questions, awesome. Feel free to put them in the chat, and I will review some of the ones that were also mentioned before, and I'm happy to answer those. And otherwise, if you want to head off into the evening, I wish you well for the week. Do check out the resources for general course information, which if you haven't looked at those, that was open since you enrolled in the course. And then do the week 1 material, which is really mostly just getting familiar with the Cornell Mushroom website and downloading some of those resources so that you're ready to go for the course for subsequent weeks. But in the meantime, thanks for your attention, and let me know if there's questions for now that I can answer. But anything we talked about tonight.